With Doubtnut, get instant video solutions to all your maths, physics, chemistry, and biology doubts. Just click the image of the question, crop the question, and get instant video solution. Download Doubtnut app today.
Hello everyone, let's do this question. So the question says the wild type E. coli cells are growing in normal medium with glucose. They are transferred to a medium containing only lactose as a sugar. Which of the following changes take place? Options that are given to us are lac operons is induced, E. coli cells stop dividing, lac operon is repressed, all operons are induced. So now let's solve this question. First of all, let's understand what is lac operon. Lac operon, it is a group of genes, it is a set of genes working under a common promoter. So lac operon, it is a set of genes working under a common promoter and are involved in lactose metabolism. Lactose is basically a kind of a sugar that is present in the milk. So lac operon is a set of genes that are involved in the lactose metabolism in the E. coli cells or E. coli bacteria. Lac operon, it is expressed. So genes of lac operon, so genes are expressed in the absence of glucose and in the presence of lactose. Because E. coli cells, if glucose is available, they tend to consume glucose more and they basically keep the lac operon turned off because glucose is their preferred food, not the lactose. So if we talk about the structure of the lac operon, lac operon has different genes that are located. These genes are the structural genes and the regulatory elements. For example, there are three structural genes that are involved in lactose metabolism: lac A, lac Y, and lac Z genes. So these are the three structural genes. Lac A genes basically codes for the trans-acetylase enzyme that basically transfer the acetyl group. Lac Y codes for permease, which codes for a transporter that allows the entry of the lactose into the cell. Lac Y, it codes for the beta galactosidase that basically involve in the metabolism of lactose. Beta galactosidase that is involved in the metabolism of the lactose. There are different regulatory elements. For example, there is an operator. Operator is a negatively regulatory site where the repressor is going to bind, and this operator basically inhibit the expression of these three structural genes. Then there's a promoter which is located upstream of the operator. Promoter is the site where the RNA polymerase is going to bind and it will allow the expression of these three genes. And upstream of the promoter is located the I gene, which codes for the repressor protein. I basically stands for the inhibitor. So this repressor protein, it binds to the operator and it inhibit the expression of these three structural genes. And there is no, but when lactose is present, in the presence of lactose, but when lactose is present, lactose basically convert into the isomeric form, which is known as the allolactose. And in the presence of allolactose, when this allolactose it binds to this repressor can no longer bind to the operator, and there's the expression of all these three genes. So basically the lactose and allolactose, they basically act as inducer of this operon, because in the absence of these inducer, these three structural genes, they are not transcribed. But when this inducer is present, it binds to the repressor, and repressor can no longer bind to the operator, and the RNA polymerase can easily transcribe these three genes. So this is a case of the inducible operon. This is a case of inducible operon, because the expression of the operon only takes place when the inducer is present. Otherwise, all the genes, they basically remain shut off. So the question was saying the wild type E. coli cells, they were growing in normal medium with glucose. So when the E. coli cells were growing in the glucose, the expression of the lactoperon is not going to take place. When they are transferred to a medium containing lactose as a sugar, so lactose basically it acts as an inducer. So when inducer is present, the genes of the lactoperons are going to be induced. So if we look at the options, the lactoperon is induced. So this is the correct answer. When the wild type E. coli cells are transferred to a medium containing lactose as a sugar, so the lactoperon is going to be induced because the lactose it acts as an inducer. E. coli cells stop dividing. E. coli cell will not stop dividing because they can use the lactose as a source of energy. Lactoperon is repressed. That is also incorrect. Lactoperon is going to be induced. All operons are induced. So this is also not correct. So the correct answer of this question would be option number 1. Thank you.
For class 6 to 12, IIT JEE and NEET level. Trusted by more than 5 crore students. Download Doubtnut app today.